Go ahead and can everybody hear me? Everybody hear me? Ben, you can hear me up there? It's good? Okay. Feel free to, there's still a couple pieces of pizza left. Feel free to come and get some if you want. It is a great pleasure to introduce Claudia Jakubzick as the speaker today. Claudia did her graduate work at the University of Michigan with Corey Hogeboom, which I'd like to say to all of our graduate students, she published 14 papers in her graduate work, 7 of which were first author. Okay, so let's get busy, people. She went from there to do a postdoc with Gwen Randolph at Mount Sinai, who is an extremely internationally well-recognized leader in monocyte biology and myeloid biology. From there, she came to actually National Jewish to work with Peter Henson, initially as a research faculty, staff, scientist, whatever that title is that nobody cares much about. But she then moved into tenure-track faculty and a multi-R01 funded tenure-track faculty, and now fully indispensable tenure-track faculty, in my estimation. Since 2010, since arriving here, she's published 29 manuscripts, 13 of which she was the primary or senior author. That's an average of 4 to 5 papers a year, not counting 2018, because we're only halfway through it anyways. And they are in notable journals such as Immunity, Nature Immunology, JX Med, Nature Communications, Blood, and Nature Reviews Immunology. She has multiple R01s, including a renewal, which is not the easiest thing to do these days. So she is a testament to the power of the force of a will, and I think what happens when you just don't sleep very much. She's been incredibly productive. One of the things that Claudia is extremely good at is taking some very basic ideas and finding all sorts of places that they belong. So the idea of monocyte biology has been, in her view and in her work, expanded tremendously out from the lung and into all sorts of places you wouldn't necessarily initially expect it to be. But it turns out all of it has significant importance. And in this particular case, it will apply to tumor immunology. So, Claudia, thank you for speaking today. I think this is okay. Yeah.
All right, thanks, Ross. That was nice. Introduction. So monocytes, actually, when I started in Gwen's lab, were thought of baby macrophages, right? They give rise to macrophages. In fact, a lot of people even thought they gave rise to DCs, but they don't. So, is it now on? Yeah. So I actually, all the data I'm presenting today is data I have accumulated in the last 6 years, of which I've presented none of it ever. And what's really interesting is, although I publish on monocytes, I've also never presented monocyte data. So I'm really happy to be able to present very little published data just in the intro, and then our current work that I hope to pull together. Okay. Because usually I present macrophages and DCs. So my lab studies long mononuclear phagocytes, mainly, and not just in the lung actually, now we're moving also into the skin because we get human skin and skin draining lymph nodes. But what are the mononuclear phagocytes? Whoops, in the lung. So you have alveolar macrophages that are behind 2 basement membranes. They seed during embryogenesis, and they just self-renew their entire lifespan. This is the same thing that's true also for Langerhans cells and microglia. Yes, Langerhans cells are actually macrophages, but with a DC-like function. Then we recently discovered 3 unique interstitial macrophages. So if you think of macrophages, and Ruslan Mechentov, who came here, gave a really nice talk about the circuitry between a macrophage and a structural cell, right? They're this accessory cell that supports the function of that structure. So for alveolar macrophages, the only reason why they live in the lung is because they're supporting type 1, type 2 alveolar epithelial cells that's unique to the lung and its function for gas exchange. On the other hand, these interstitial macrophages are present in all organs we've examined. And that kind of makes sense too, because all organs share a common glue, right? Endothelial cells, fibroblasts, smooth muscle cells, lymphatic vessels, etc. So whatever they're doing there, they're probably behaving as similar accessory molecules there, and their transcriptome is almost identical. And then there are two DC subtypes, and this IR4DC, I mean, I named them based on the transcription factors that promote their development, which you'll see in a bit, but I'm actually presenting almost no work on DCs. So it's all about monocytes. But anyway, these are the dominant cell types that are thought of as antigen-presenting cells in the draining lymph nodes. And for whatever reason, monocytes get ignored a lot, especially in the DC world, and in other worlds, but not in the cancer world. So, so here we have monocytes. So how did we come to realize that monocytes were not baby macrophages? I mean, I did extensive lineage studies with a lot of lineage mice, but that's all in this manuscript here. I would say that this particular data is probably the most powerful data of all to demonstrate this. So if you pair BIOS mice, you get a 0.1 and a 0.2 congenic mouse, and I kind of like walking around, but I guess I asked for this. You cut it along the side and then sew them together. They'll share circulation within 4 days. You'll have almost 50-50. And then and here you can look at 2 weeks, but it doesn't matter whether it's 2 weeks, 6 months, or a year. What you're going to find is this. So if you look at lung monocytes, they're about, actually they never reach 50-50, monocytes don't, but they're about 60-40. So here's the host, and here's the donor monocytes within the CD45.1 pair, and then here is the host and the donor monocytes in the CD45.1 pair, sorry. And so what you see is if you focus on the macrophages, you only find them from the host. So what this clearly says is that monocytes are not continuously replenishing macrophages. So let's also then think about monocytes in the circulation. So monocytes have about a half-life of 20 hours. They are the most, in both human and mice, abundant mononuclear phagocyte in circulation. So if they have a half-life of 20 hours and they're not giving rise to macrophages, tissue macrophages, then what are they doing? They're that abundant, and they never reach 50-50 circulation, so they're not either staying in the circulation for extended periods of time. They exit the bone marrow and go straight into tissue. So what they're doing is they're constantly surveying the environment. And surveying for what? Who knows? Now, I'm not saying that monocytes don't give rise to macrophages. They do. And if you have a niche that's open, they replace it. If there's an inflammatory process, they can become macrophage-like and DC-like. But in your everyday life, what are they doing? Okay. So how do we identify monocytes? I hope everyone's fluent in flow. All right, in the lung, because I studied lung, but it can be any organ. You can't do CD11C versus CD11B because it just gives you a gamish of cells. So we ended up doing a transcriptome analysis of all the mononuclear phagocytes, and what we identified was the co-expression of MERTK versus CD64 selectively identifies macrophages. So here you can see alveolar macrophages and the 3 interstitial macrophages, of which I'm not going to discuss today. If you take this macrophage negative gate and then replot as CD11C versus MHC class 2, you can see here the 2 overarching DC subtypes. So you got to remove a lot of cells to identify monocytes. And then if you then take that and then go down, you can either use CD64 or you can use F480. These are eosinophils, these are neutrophils, and you can find monocytes. It turns out, though, in steady state that most of these are in the intravascular space. So if you're going to study monocytes in any given tissue, oh, I was doing my next slide. Sorry. Macrophage DC, I need to focus on this side. I'm telling you, I don't do this. So DCs, and then here are the 2 DC subtypes, and you take this macrophage DC negative gate, you can do CD64 or F480, where you see eosinophils, neutrophils, and then you can take these monocytes, and if you're going to use the F480 clone, by the way, the only clone that works for staining monocytes, not macrophages, is the original clone from Simon Gordon, which is on my next slide, if you want to jot that down. But anyway, and then most of these monocytes, as I was saying, are in the intravascular space. So if you're going to study monocytes, especially in steady state and in inflammation, or neutrophils for that matter, you should always do an IV anti-CD45 right before death, because it's hard to distinguish the two. So how do we identify it in blood? So this is actually just one mouse, okay? You gain on this intermediate side scatter, CD115, and then when you plot Ly6E versus MHC class 2, what you can clearly see is that there's a lot of populations here, right? However, this is one, and this is another. And these Ly6E low monocytes are actually the vacuum cleaners of the luminal space in your blood vessels. They don't ever enter tissue. And the reason for that is because these Ly6E high monocytes, when they enter into circulation, they have very high expression of L-selectin. And L-selectin is absolutely required for its entry into tissue. Now, if in the pure knockout, it can somehow use other molecules, right, other transmigration molecules. But if you put it in a competitive setting, like if you were to transfer 50-50 wild type and L-selectin knockout, you will find no monocytes derived from the L-selectin knockout in tissue and lymph nodes. And as you can see, the Ly6E low monocytes don't have any L-selectin and other transmigration molecules because they're the vacuum cleaners of your intravascular space. So, one thing to caution is that when I find that people say Ly6E low monocytes enter tissue, what you need to realize is that they actually were Ly6E high classical monocytes that enter tissue and start downregulating Ly6E once they interact with the endothelium and upregulate MHC class 2 and other antigen presenting factors. Okay, so then in a draining lymph node, this is a mesenteric lymph node or skin draining lymph node. You can get CD11B, try and avoid the DCs. You can do side scatter. Here are eosinophils that are F480 positive. And then what you can see is these are the monocytes that just came in, and as they enter the lymph node, they start upregulating MHC class 2. And you can see them in both settings. And not just MHC class 2, CD206, CD80, etc. So how do they enter lymph nodes, right? So like DCs, that there's two routes of entry. So you can come in through the HEV, and this term is called lymphoid DCs, or you can come in through afferent lymphatics, which are termed non-lymphoid DCs or migratory DCs. Monocytes can do the same, except that they're indistinguishable, unlike DCs. So I don't know if they're different or not. But what you can see here is in a wild-type mouse, you find monocytes. In a fractalkine receptor knockout mouse, you also find monocytes. And I thought that you may not find it because one feature of monocytes is that their fractalkine receptor really high. And a fractalkine receptor is a chemokine receptor, but they don't require for entry or differentiation of these monocytes. Now, what they do require, not only for exit from the bone marrow, but also entry into lymph node and tissue, is CCR2. That's a chemokine receptor that monocytes really require, but DCs do not. And I'll, again, show you data of that in a bit. Now, so L-selectin, as I said, is required for entry in through the HEV. But you can see that here we still have monocytes, but in these mice, data I'm not showing, but has been shown by others, is that they lack lymphoid DCs. The PLT mice are CCR7 ligand deficient mice. So you don't get migratory DCs in the CCR7 knockout. You don't get migratory DCs because you need CCR7 and CCR7 ligands to migrate down afferent lymphatics and enter tissue. But you can see that monocytes can enter through the HEV, but they lack migratory DCs. How do we know monocytes require, oh wait, before I get there. The other thing I just want to show you, because you're going to see once I get to the hardcore data, I use the CCR2 knockout mice a lot. So as you can see here, monocytes do not readily enter tissue in CCR2 knockout mice. You can find it in wild type, but you can barely find tissue, if any, monocytes in a CCR2 knockout mouse. So how do we know it requires CCR7? I thought this was a pretty cool study I did about 15 years ago. And haven't published it. So this is CD11B enriched for either 0.1 wild type cells or 0.2 CCR7 knockout. And you mix it once you've enriched it 1-to-1. And then you transfer it into the skin, and you can see 24 hours later we can find them You can find the cells that were adaptively transferred. But in the draining lymph node, only the wild type cells migrated that had CCR7 expression, whereas the CCR7 deficient DCs and monocytes didn't. So you need CCR7 for monocyte migration, and we ended up sorting them independently and so forth and showing the same. How do we know monocytes traffic to a draining lymph node? In this case, this was a bit indirect because they're few and they're very hard to see. But here we gave an intranasal bolus of anthrax spores, which causes a ton of monocytosis in the lung. And I don't recommend tracking cells this way, but it was the way we tracked it. So we gave through the intranasal route CFSE, which what happens is it's so reactive, it labels everything that's in that environment. And then we looked 24 hours later to see what migrated to a draining lymph node. If we gave purely on myeloid cells, what we find is that what migrated to a draining lymph node were monocytes and migratory DCs. So how about turnover? I mean, not just, a lot of work has been done actually by Simon Yona and Stefan Young about monocyte turnover, but we also did some with BRDU, but here I thought this was one of the most striking pieces of data, again, that also hasn't been published. So if I IV inject clodronate, which is also a pretty messy system and way to deplete monocytes, and don't recommend it, but if I IV inject it, what you find is 24 hours later, you lose your monocytes from circulation, right? But if you look at the mesenteric lymph node or skin draining lymph nodes, and the reason why I have this population here is you can see the DCs are there and they're just fine. But the monocytes disappear within 24 hours, and that's again a testament to their turnover. And it's not because these monocytes are the ones in circulation, because you can IV CD4 T and you can find that all these cell types are actually within the lymph node. So what it says is that these monocytes are constantly trafficking and moving through. So CCR2 positive, Ly6C positive monocytes constitutively enter tissue and lymph nodes without full differentiation to a macrophage or DCs, though they induce MHC class II and express other markers of MO-DC phenotype, like DC sign, which I'm not showing here. And then entrance into lymph node can either happen through the HEV or afferent lymphatics. And it goes together with the upregulation of MHC class II, CD206, and other co-stimulatory factors. And this becomes relevant when I show you some human data. Okay, so what are the lymph node APCs? You actually have three. So you have these BATF3 positive DCs that are really good at cross-presentation. I personally have not been able to see them load peptide on MHC class II. I've tried many different transgenic models, but people have published that they do. I think it's more if you feed the peptides in vitro, then they can just freely load. But it kind of makes sense that they don't really present to MHC class II, because what this cell type does is it engulfs exogenous antigen and instantly releases it into the cytosolic compartment. To actually load up on MHC class II, that endosome has to fuse with the lysosome to allow that loading to happen. And it just doesn't happen in the BATF3 DCs. Then we have Ly6C positive monocytes, and our 4 DCs are really good at holding on to that antigen and loading on MHC class II. Now, here I put pluses everywhere, because the point of this review was to talk about monocytes as antigen-presenting cells, and I really tried to focus in this review article on papers that study monocytes in vivo, not take them out and then throw them in a dish. And the reason for that, too, is because if you take blood monocytes or splenic monocytes, what you're mainly going to have is these immature monocytes that really require signaling and transmigration by endothelial cells to become the cell type that you're trying to investigate, that antigen-presenting cell type. So it doesn't really work if you just plop them into plastic. So there wasn't that many papers, and a lot of it's kind of induced, but it seems like they do everything. And a little bit of a testament to that is that there are 2 papers that recently came out a few months ago from also Stefan Young's lab and Elodie Segura, who what they did was they did single cell analysis on both human and blood monocytes in circulation. And what they found was an incredibly homogeneous population. So what this says is that as precursors, what they're probably doing is entering tissue and getting ready to get the signals that are required to become whatever it is that they need to become, a TIP-DC, an alternative macrophage, inflammatory monocyte, et cetera, MO-DC. It's a bit of a name your flavor. So what is a monocyte? So the question I had is, whoa, what is the monocyte contribution in adaptive immunity? So when we published this paper that monocytes are constantly surveying the tissue and they're not becoming macrophage or DCs, clearly that's the next question that came to my mind. And so in this period, it was a hot time for resident memory T cells. And so there was this model, that showed that if you gave vaccinia virus intradermal in one ear with ova, you get these resident memory T cells that seed there, but they don't seed in the other ear. And since monocytes are constantly surveying the environment, I thought maybe they're providing survival signals, retinoic acid, differentiation signals, et cetera, because they're the ones that can move everywhere. So it seemed like a pretty logical thing, right? So, and people were saying how it's not just regulatory T cells that need retinoic acid for its differentiation. All effector T cells need this, and monocytes express these enzymes really well. So with Ross, and we did several studies, we gave OT1 T cells. He inoculated through the intradermal route the vaccinia virus that expresses ova. And then 30 days later, I harvested skin and skin-draining lymph nodes. And okay, so the hypothesis is that you need monocytes for resident memory T cell survival, okay? That's the hypothesis. When we looked at wild-type mice, and he inoculated the right ear, I couldn't really find them, nor in the cervical lymph nodes. I mean, very few. And this, we did a bunch of mice. So, and if you look at the CCR2 knockouts, you can find it in the right ear, not the left, and you can find it in the cervical lymph nodes. So this was complete opposite of our hypothesis, right? So now I had to then restate our findings because we performed this study 3 times and gave an alternative hypothesis, which is that monocytes dampen the induction of a cytotoxic T cell response, and it's only recently that we are starting to recognize that it's through the induction of suppressor CD4 positive T cells. So how does this start? So let's look at wild type and CCR2 knockout mice. So one thing is to induce a cytotoxic T cell response, and in the case of the lung, the major player and probably the only player in this context is the BATF3 positive DCs because of poly-AC and how it expresses TLR3 and so forth. The major player is BATF3 DCs. So what you can see is here in the wild type lung draining lymph node, here are your monocytes. If you gate on DCs, here are the 2 DC subtypes. But if you look at a CCR2 knockout mouse, you don't have many monocytes. These are actually a few neutrophils. And then, but if you look at the DCs, they appear fine. And it's whether the numbers are roughly the same, whether it's soluble ova or soluble ova plus poly-IC. So then we looked at what happens then in the absence of these monocytes. So this is a model system that I use from here on out. So you transfer naive OT1 T cells that are CFSC labeled into either wild type or CCR2 knockout mouse. You give through the intranasal route soluble ova and poly-AC, and then you analyze the lung draining lymph node 3 days later for interferon gamma and TNF alpha production. So really cytokine production. And what we find is that in the absence of monocytes, we had this huge increase in cytokine production and TNF alpha. And we really didn't have that much of a change in the number of CD8 T cells that we would observe. So now this brings us to the question of what is the mechanism? Oh, sorry, I'm going ahead of myself. I just put this together 4 days ago. So of course, you know, one caveat to the CCR2 knockout is that someone says, well, it's a CCR2 knockout and the mice have lived like that forever. So what we did is we depleted lymph node monocytes using anti-GR1. Now, anti-GR1 doesn't just deplete monocytes. It's not my favorite antibody. It depletes PDCs, it depletes some T cells, but it also depletes monocytes. And when we depleted with anti-GR1, we still see this increase in cytokine production from the OT1 T cells, both in interferon gamma and TNF alpha. We also did the anti-CCR2 antibody, which is the MC21 clone, but I just have, it's too few numbers to say anything, but again, it also trends the same as the anti-GR1. So to evaluate the mechanisms by which monocytes dampen cytokine production, we created chimeric mice. And so you can take a 1-2 mouse or a 1 or a 2. You just then have to have good controls. And then you can transfer 8 to 2 or 80 to 20, CCR2 knockout bone marrow and either as a control wild type CD45.1 or your experimental knockout mechanism that you're investigating. And what results is that 100% of your monocytes that enter tissue, because it requires CCR2, derive from this donor bone marrow, the knockout or the wild type. So let's look here. In this case, I'm doing the mediastinal lymph node, but later on I'll show you how to knock all the APCs out selectively. So what you can find here is that in this case, I have to use the 0.1 always as a control because my knockouts are all 0.2. So here what you can see is that if you gate on migratory DCs and make sure you don't have monocytes or macrophages there, and because this is a mediastinal lymph node, you also have this other DC that is very specific to the gut, this double positive CD103, CD11B. It's not dependent on BATF3. And then monocytes, what you find is that it's only the monocytes that derive 100% from the CD45.1 bone marrow donor, whereas the others don't. They're 80-20. So when we do that, what we look at is, first we wanted to know, are monocytes interfering or competing with the cross-presentation of the BATF3 positive DCs? So what we did is we acquired the BM1 mice, where they have mutations in the binding site of MHC class 1, doesn't allow them to load Symfecal. We also acquired the BM8 mice from Ross Kettle. We had similar observations, and what we can see is that there's no increase in interferon gamma or TNF alpha if the monocyte isn't interfering by presenting Symfecal itself. So then we asked, well, what about PD-L1, right? And the reason why is because in the immunity paper where I showed that monocytes are surveying the environment, one of the 15 genes that went up in the tissue from blood to tissue was PD-L1. So PD-L1 is what we inhibit for the immune checkpoint blockade, and so we thought, let's look at that. So I called Dan Barber from NIH and asked him for mice. He said he only has 1-2 mice. Monocytes don't express 2, but they express 1. And what you can see here is when they come into the lung, if there's no inflammation, they have very low level expression, although it can be detected by the transcriptome. But in the presence of poly-IC, they have a ton. And in the draining lymph nodes, they also have a ton, like DCs expressed. So this, I'm going to come back to this because this bummed me out. So when we deplete PD-L1 expression on monocytes, what we saw was a decrease in interferon gamma and TNF alpha expression. Not an increase, not the same, a decrease. So mind you, when we did this data, and this is not the only time, several times, at this point, my hands are up, I'm like, we don't have a story. I don't know. Because by this point, we had already looked at TNF alpha, INOS, BM1, BM8, and PD-L1 too. So the only story is that in its absence, we have an increase, but the mechanism, I don't know. And we even tried to figure out whether it's due to lymphatic endothelial, lack of lymphatic endothelial cell development, and we really couldn't see a difference, but maybe with the help of Beth and her expertise, we might have. Okay, so a question that would come to mind is, how about FOXP3 cells? Are FOXP3 cells different in the wild type and the CCR2 knockout mice? And because I hadn't summarized this data till about 4 days ago, I didn't realize every year I asked that question. So I have over 20 experiments looking at the presence or induction of FOXP3 with either the antigen or poly-IC. And whether in numbers or percent, we never saw a difference, at least not natural Tregs. And with CD8 T cells, I mean, this is the way I can divide it into 3 populations, and then there's others. We didn't see much of a difference there either. So at least for natural Tregs, it doesn't appear that they're playing a heavy role, at least in frequency of number and percent, although I understand that antigen specificity is more important. So the next low-hanging fruit and the most boring of all, because at the time everyone knows that IL-10 suppresses immunity, and I was really hoping that it was something different, but it turns out that it's IL-10. So monocytes entering tissue significantly increase IL-10, right? So we acquired the IL-10 reporter tiger mice from Jackson. And what's really surprising for me and striking, but not too surprising when I look at the transcriptome data, is that it's only monocytes that are expressing IL-10. So here's an example in the lung. What you can see is when you give soluble OVA, we have some IL-10 expression. This is just taking straight out of the lung and going to the fax machine. And most of those monocytes, I said, in like steady state are in the intravascular space. So it's monocytes that are interacting, Ly6E high monocytes within that intravascular space of the lung that are expressing IL-10. But if you add soluble OVA, what you see is monocytes come in, not only do they upregulate MHC class II and CCR7 and CD206, et cetera, they also upregulate CD64. And what we find is that the CD64 higher, the ones that are higher are in the lung, and then there's also the intravascular monocytes. And what we find is that it's only these cells that are expressing IL-10. Okay. And what's really interesting is we're actually doing studies now where we don't believe that it's a direct effect because poly-IC does not hang out, especially 10 micrograms of it when you give it through the intranasal route 3 days later while these cells are constantly migrating in. But yet these cells are all upregulating IL-10. So what it tells me is that there was a lot of inflammation, and it's kind of a uh-huh and a duh moment at the same time, right? They come in and they're there to put on the brakes. I mean, in the T cell world, we have Tregs and effector T cells or suppressor T cells. So there's always a yin and yang. So why wouldn't that also exist in the APC world of different cell types? I mean, there's this idea that an immature DC promotes tolerance, whereas a mature DC doesn't or something. But why isn't there better, like just a division of labor between these APCs? So, okay, so now we created mice where the monocytes selectively are depleted of IL-10, and we see an increase in interferon gamma and TNF alpha in these mice. So it turns out that it's IL-10. And we've just recently stumbled into these findings. It took us a really long time to get here because I was kind of ignoring the data for a very long time, but I'll tell you what brought it back, and it's a human study with poly-IC and tumor antigens. So this comes to mind that, wow, now studying what monocytes do is important. And it's also because, as you'll see in a minute, humans have tons of monocytes in the draining lymph node. So to think that they're negligible and not doing anything is wrong, right? So then now this was data just acquired this last month. We have some preliminary data where we ended up creating, we crossed the CCR2 knockout to the IL-10 promoter so that we have a CCR2, not promoter, sorry, reporter. And so we have a CCR2 knockout mouse that reports IL-10, and we have a wild-type mouse that reports IL-10. And in that setting, what we find when we induce the IL-10 production in an antigen-specific way, we find more IL-10 antigen-specific T cells compared to the CCR2 knockout. But again, this is preliminary data. These mice are coming, and we're going to keep doing research as they come through. The other thing that we did was in vitro, we plated OT1 T cells with either APCs, so just CD11C positive cells from the spleen with OVA. And when we add naive CD4 T cells, we have an increase in interferon gamma and TNF production. When we add CD4 T cells from wild-type mice that went through the process of antigen and poly-IC, we see a decrease. But when we add it from the CCR2 knockout mice, we see an increase. So what this suggests is that CD4 T cell that would be present in wild-type mice that suppresses the OT1 T cell induction cytokine production is not there in the CCR2 knockout mice. So why is this interesting? I mean, why is it interesting to study poly-IC and antigen and the role of monocytes in its induction in effector CD8 T cells? So this is what brought back all this data. So this really elegant Nature paper, although I'm sure there's many, many others, and I'm not exactly sure how I stumbled into this, did these studies, and they showed 100% efficacy. So you take, what do you do here? You take tumors or tissue from the same patient, and you do a DNA and RNA sequencing analysis to find the specific mutations. You run it through a whole bunch of algorithms to the likelihood of the peptides that are being presented. And what they did was they synthesized somewhere between 50 to 100 peptides in the presence of poly-IC and injected the patient, the melanoma patient. And they did these primings and boosts. And what they found was if you were stage 3 melanoma patient, you had 100% remission. So these people survived. With the stage 4 one, though, they used anti-PD-1 and they had then 100% remission. So this then made me think of this PD-L1 data, right? What was going on here? And why, when you give these immune checkpoint blockades, specifically PD-1 or PD-L1, do some people survive and some people don't? And then again, what's the role of monocytes in this process, right? So, as I said, when we depleted PD-L1 from the monocytes, we had this decrease in cytokine production of the antigen-specific CD8 T cells. So what we believe is happening is this yin and yang effect, okay? We have these two immunogenic DCs that when you block it, you have an increase in a cytotoxic T cell response because it doesn't put the brakes on the cytotoxic T cells that it's differentiating or priming. But on the other hand, and we know that monocytes are not directly presenting the antigen to CD8 T cells, right, because of the BM1 and BM8 studies. So when we block the monocyte from PD-L1, what's probably happening is we're blocking its ability to regulate the suppressor T cells that it's producing, and therefore they're proliferating out of control. And so when you have more of these antigen-specific suppressor CD4 T cells, you'll have a decrease in a cytotoxic T cell response. So that's what we believe is happening. I don't have the data yet, but because there's lots of students here and if you want to study APCs, I thought I would show you how do you ask that question in vivo. So what we've created are mice where either the BATF3 DC selectively, 100% of them are deficient of PD-L1, where either the IR4 DCs 100% are selectively depleted of PD-L1 and 2, but not the other APCs, or when the monocytes are 100% selectively depleted of PD-L1 and 2. And the way this data looks like, and this I also just got last week, is like this. So this is looking at a skin draining lymph node because I'm harvesting the lung draining lymph nodes for other things. But this is also true for the mesenteric lymph nodes. So if you find DCs, and you have CD103, IR4 positive DCs, or monocytes, and then now this looks noisy, but just follow me. So the top row are gated on only BATF3 DCs. The middle row is gated only on IR4 DCs, and the bottom row is gated only on monocytes. This half is bone marrow chimera that's mixed with wild type. And this half is bone marrow chimera that's only mixed with PD-L1. So the first one is a mixture of BATF3 bone marrow with wild type 0.1, and what you find is that the BATF3 100% derive from the other bone marrow. In this case, it's the IR4 bone marrow mixed with wild type 0.1, and what you find is that the IR4 DC 100% derive from the other bone marrow, but the BATF3 DCs derive from both, and so do the monocytes, right? And so then, and in this one, you have 100% of the monocytes derived from the 0.1, but the BATF3 DCs and the IR4 DCs derive from both. You can also look at this with PD-L1. So the PD-L1 was the donor bone marrow. So here, the BATF3 DCs don't express PD-L1, but the others do. Here, the IR4s don't express PD-L1, but the others do. And here, the monocytes don't express PD-L1, but the others do. Or maybe it's this way. Sorry, I'm kind of... Oh, sorry, CD103 this way. So it's this way, and this is a mouse. But you can see monocytes don't express PD-L1, but IR4 DCs and BATF3s do, and so forth. So now you have a way that if you want to study these cell types in vivo, you can create these bone marrow chimera systems where this works. And if there's a gut person here, the IR4 positive DCs bone marrow is the way to go. So why is it interesting? Again, another one is because, like mice, in humans, lymph node and skin draining lymph nodes, there's many monocytes as there are DCs. And in fact, now we are doing both single cell analysis and RNA-seq bulk, where we're aligning human monocytes and mouse monocytes. So both transcriptionally, functionally, turnover, it's the ones that align best between monocytes and humans. And what's really cool is a friend of mine, bless his heart, he looked at human monocyte turnover and injected LPS through the intravenous route because it causes depletion, and then tracked to see how long it takes for them to come back. And published this really beautiful paper in JEM. So he showed that the turnover of both human and mice is about the same, about 20 hours. All right, so I'm going to show here a PCA plot of the human, because what you can see is that blood and lymph node, or in this case, and lung monocytes and blood monocytes, all cluster very closely together, whereas depending on where you extract the macrophage, it can be all over the place. So in mouse PCA plots, if you get monocytes from regardless of what tissue it is or what lymph node or the blood, they all kind of cluster together. So the changes are minor if you have other cell types in comparison. So here's a picture of a human lung draining lymph node. And what you can see is that the human lung draining lymph node is different from the mouse. So this CD11C, sorry, got omitted out here, HLA-DR. These are neutrophils. They're not PDCs. These are monocytes, and these are your DCs. So if you take side scatter a certain amount, you can find that you can identify DCs. So if you want to target a DC and induce immunity, you should use CD1C and DEC205. The DC1 and DC2 that people find in blood does not exist in tissue. I have skin draining lymph nodes, lung draining lymph nodes. It does parallel beautifully in blood and mouse, but those blood DCs that you find in human does not parallel with tissue human or lymph node human cells. And then what you can see here are monocytes. But just to show the appreciation of monocytes a little more, this is another human lymph node. I've done this over 100 times. And what you can see is that here are monocytes, here are DCs, here are CD141, which normally you think is DC1, but it's not. It's monocytes. It's just thrombomodulin. When a monocyte enters tissue, it upregulates CD141. CD64, CCR2, it starts upregulating CD206, and then these are DC markers, CD36, CD11B. You can see on and on and on, both at phenotype, both on cell surface or transcription, it looks a lot like a monocyte. So, and you can also appreciate how abundant they are in the lung draining lymph node of a human, and also this is again true in skin draining lymph nodes as there are in DC. So they're not negligible cell types. So what do they look like? I was saying that a monocyte really needs that endothelium to change, to become the cell type that it's going to become. So we have a very strong appreciation for intravascular delivery of anti-CD45 in a mouse, because if you perfuse a mouse white, it looks like this, all you've done is you've pushed through those red blood cells, but you've left behind a ton of leukocytes within that intravascular space. So if you just digest that lung and assume that everything's in the tissue because you perfused your lungs, it's not true, because these lungs were white. So what we did is we grabbed the human lobe and we did the same thing. And it was more for me to identify what's intra versus extravascular mononuclear phagocytes, but I'm presenting this data because I think you can appreciate the change that happens to the to the monocytes that enter tissue in this particular analysis. So what we did here is I gave 4 complete vials of anti-CD45 and per-CP through the intravascular route, the pulmonary artery. I mean, this lobe is huge. It's like this much, 150 cc's. And then through this route, through the intravascular route, I put in 4 anti-CD45 FITC vials through here, another 150 cc's, and I clamped all of this and I held it for 20 minutes massaging this lobe. I did this 8 times, okay? And so then you cut out, then you wash it all out, and I washed it. I mean, you push it through, and then you analyze the data. And so what you find here is these are the cells that were in the intrabronchial route and the intravascular route. And what's cool about this is now it's a single cell suspension, so it's free for all antibody binding. You're not comparing 2 separate samples, and maybe you gave a little too much more antibody here or there. It's free for all, right? And it was exposed the same way to digestion enzymes, etc. And I just want to point out what you can appreciate here is that you have the bronchial route staining or intravascular route. You can see lots of neutrophils in the intravascular, not so much in the intravascular. You find CD11B, CD4 T. If you didn't do this, they would be together. But then as you move along, those that came into the tissue upregulate MHC class 2, CCR7, CD1C, CD206, etc. So actually the point of this data was to say that most of us won't be able to do this with human tissue. So if you really, I was really trying to find a marker that can really tell people whether their mononuclear phagocyte is intra or extravascular. And that molecule is CD206, with the exception of DC1. DC1 does not express CD206, but every other mononuclear phagocyte, when it enters tissue, it expresses the mannose receptor. But in the intravascular system, it doesn't. So you won't find any mannose receptor expression or monocytes or DCs in humans in circulation. Okay, so we ended up sorting all these cell types. We've sorted over 10 different individuals. Here we're showing our first set, and what you can see is that DCs will cluster together, blood DCs, lymph node DCs, lung DCs. Macrophages kind of spread out, but monocytes, whether it's blood, lung, or lymph nodes, all kind of cluster together. And so, for the sake of this purpose and this talk, we have, I have a ton of data, but it's so overwhelming, and really our bioinformatics team at National Jewish Health is trying to crunch it together to find its true orthologs and human and mice. But I thought what would be fun is to just show you IL-10, because this point of this talk is about monocytes and what aligns with human and mice. And what we find nicely is that in the blood, the CD14 positive monocytes don't really express IL-10, but in the lung and the lymph nodes they do, and then the lymph node DCs do not, and neither do the blood DCs. And so, and of course, these are monocyte signatures, MAFB, CCR2, blah, 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 and these are DC signatures, ZBTB46 and CCR7. So with that, I conclude that monocytes, what they appear to be doing, at least in the context of poly-IC, and because we're using this as a therapy, cancer immunotherapy, we should really think about the ratio in numbers and what kind of role are they playing, and should one deplete them? I mean, you can deplete them while you're giving this therapy to really give that booster effect, right? But they put the brakes on the cytotoxic T cell response, and it's through the production of IL-10, and I believe it's indirect. So it's based on the induction of some suppressor CD4 positive T cell that expresses IL-10. I don't know if it expresses FOXP3. We're really trying to figure that out. And more so, the relevance of this study is because monocytes of any other mononuclear phagocyte, besides your classical macrophages, best aligned both in human and mice transcriptionally, functionally, turnover, etc. So with that, I end by acknowledging really Sophie and Miglena, who did most of this work. Of course, there's other members in my lab who are no longer with me, but who dabbled in here and there. Hopefully I'll have this cool transcriptome data coming soon. Sonia and Thomas are the leads in this as the bioinformatics team. Let's see. And then, it just wants to quit. Is it there? Oh yeah, it's there. Okay. And then Ross, who really, I don't know how Ross starts all my studies. Like I have most of my papers, it's like I'm talking to him and he says something or does something and I'm like, oh, that's interesting. So he's a great person to talk to and he sparks ideas. And then Eric Clamby, who helped me a lot initially with all the T-cell biology. Okay, and with that, that's it. Thank you.
Does this work? Hello? Check. Okay, good.
So one comment, one question. One, this idea of PD-L1 knocking it out and having a reduction in interferon gamma and in the CD T cells. Well, it's unusual. It's not unprecedented because I think Mark Slomchek has described in his NI paper that when you knock out PD-L1 off B cells in the germinal center, that the TFHs don't spit out as much IL-21. And so there's some idea of this PD-L1, PD-1 interaction sending something more than besides the inhibitory signal, which I think is unusual, but it's there already.
Yeah.
But the question is, so in monocytes, there's this non-classical population. I think they're NER77 driven. They're supposed to be involved in early response of the monocytes into the lungs, for example, to eliminate a lung cancer. Do you know if those are also are those also big time IL-10 producers?
Big IL-10 producers?
Are they the non-classical ones that are supposed to be involved with tumor elimination? Are those?
Right. I don't see IL-10 production out of them.
Okay.
No.
So I have two questions.
Yeah.
So at the thing that started this out was a CCR2 knockout and increased resident memory cells,
Yeah.
right? So to circle back to that,
Okay.
do you think the antigen is still there in the ear? Part of the job of a monocyte is to go in and clear out antigen reservoirs. Antigen persists at times. So do you think it's still there and that's one of the reasons for that?
I don't know. What do you think, Beth?
We ever tried that? No? No, you haven't done it, or no, you don't think so?
That's yeah.
So you're asking if the monocytes are clearing out the residual antigen?
In the absence of CCR2, do you have a buildup of antigen within the tissue?
I think
A maintenance of antigen in the
I think
tissue?
in the infection model, maybe.
Yeah.
But in like this 1 microgram of ova poly-IC, yeah, right. So macrophages, and you have the subcapsular sinus macrophages, and it's just such a low quantity of antigen. I'm not sure that.
Okay.
But maybe in the vaccinia virus, it's different, right? Because you need probably monocytes to clear that virus. So it could be that the virus itself is persisting.
So the corollary to that is the other version is if it's not that, then there's something about IL-10 that's inhibiting resident memory formation, right? So if monocytes are there making IL-10, is that what's... So if you had IL-10 deficient monocytes, would you recover? And
I don't know. It's a fascinating question. I don't know.
Okay.
I don't know the answer.
And then last question. It turns out I have three. The PD-L1 blockade did the opposite you expected, right? And so the simplest answer is that if a monocyte doesn't have PD-L1, it actually makes more IL-10.
Oh, okay.
Right? So if you put the reporter onto that, which of course is another however many years of breeding, or just look for IL-10, right? That'd be the simplest answer, is that it elevates its IL-10 production and it squashes the T cell response.
But yeah, it's possible. Okay. It's possible. I think the suppressor T cells is just one mechanism. I mean, there's probably a host of mechanisms. It could be a dominant mechanism in this particular setting. I do believe lymphatic endothelial cells are playing some role, because part of the idea that, again, all structural cells need an accessory cell, and monocytes are the one that are abundant in that T cell, B cell interface, and that's where the lymphatic vessels grow. And when you have inflammation, monocytes start expressing the proper growth factors. So but our technical abilities didn't really show differences. And this is why it would be good to collaborate to see if that's also a contributing factor to why we see this enhanced response.
So Steve Dow's group, and also we have seen that with the PD-L1 antibody blockade, that actually it binds to either macrophages or lymphatic endothelial cells and helps them to survive better. So I wonder if maybe this is like you're having differences in the ability of the monocytes or any cells that you're looking at in regards to PD-L1 and their ability to live or die or divide, or have you looked at those things yet?
In a PD-L1, no. So I have another piece of data that's really interesting, and I can't explain. So since we're talking about this, let's see if I'll find it. It's probably in here. My first, I decided not to put it in. Let's see. Sorry. Oops, I didn't mean to do that, but okay, well, I think it's here. So one of the things that, and this is again, comes back to the lymphatic endothelial cells where I'm not sure what's happening. So Shannon Turley, right, who studies lymphatic endothelial cells, and this is another reason to do TNF and INOS and so forth, demonstrated that after an inflammatory induction, these lymphatic endothelial cells deplete T cells by the production of nitric oxide. So we thought that perhaps if the monocytes can promote the development of those lymphatic endothelial cells, then you can't shut down the T cells. But it's not really the number of T cells. But anyway, what you can see here is that in these mice, with our model, right, the soluble ovum poly-IC, either at day 1, day 3, or day 5, the wild type mice, what is this? Sorry. Again, it's like constantly reasoning, right? They don't turn over as readily as the DCs from the CCR2 knockout, right? So this was a little counterintuitive for me because I thought it would be, what was it? I'm sorry, right. Anyway, sorry. I know I had a reason for this, but it's like escaped me. Anyway, the point is that the turnover rate of the DCs in the draining lymph node of either wild type or CCR2 knockout mouse, it's much more rapid in the CCR2 knockout mouse than is in the wild type mouse, which again for me was a little counterintuitive because I thought that the DCs would stay to present the antigen a little longer in the CCR2 knockout mouse compared to the wild type mouse. So the fact that the turnover rate of the wild type mouse DCs is greater than the CCR2 knockout is again a bit confusing for me. And so I'm trying to find a way to rationalize this data. But that's why I'm saying, like, I think IL-10 is just one contributing factor in the suppressor T cells of the induction of interferon gamma. It's a pretty dominant one, but how it's affecting the overall environment, you know, is unclear. But at least the IL-10 production from the monocyte is a dominant factor. Yeah?
Claudia, you talk about the monocytes as being the vacuum cleaners. So what do you know about the phagocytic capacity of the amount and the fate of the cargo?
So the vacuum cleaners, I was talking about the Ly6C low monocytes that remain in the intravascular space. And I actually know a ton about that because we used to give anywhere between 0.5 to 2 micron beads IV, and it turns out that Ly6C low monocytes are capable of really acquiring these cells because they're adherent to an endothelial cell. So because they're adhered, they can catch these beads, you know, running through circulation. Whereas the Ly6C highs, because they're not crawling on the luminal side of the endothelium, you know, they don't pick up the beads because it would have to hit them in the face, right? And they're also constantly trafficking through. So the size, it's pretty high. They have a lot of phagocytic receptors. They can pick up dying cells readily. So yeah.
And if those are not beads, but are organisms or various things that are going to result in signaling inside the cells, then what does the cartilage, which is the fate of the monocytes?
Oh, that's interesting. So are we talking about monocytes in circulation or monocytes in tissue? So Ly6C low monocytes are relatively, in a monocyte world, long-lived. And that's because they're differentiated macrophages of the blood. And it's hard for macrophage biologists to hear that because they think of macrophages as these giant cells, which is what they are. But in the blood, they can't be that giant. So, okay. But in tissue, if there's inflammation, then a monocyte can differentiate into a macrophage and can gain all the parameters of hallmark features of a macrophage, and they can persist for the extended period of time that that inflammatory process is there. But what people find is that there's two waves, right? And it probably depends on the signals that are happening when a monocyte enters. There's a pro-inflammatory wave of the macrophage that's required for clearance and digesting and killing, and then there's a reparative phase where that monocyte is coming in to be something else. I don't think the tissue macrophages itself are playing that dominant of a role in those pro-inflammatory and anti-inflammatory functions, because they need to support the structural cells that promote the survival of that organ or the function of that organ. So it seems like monocytes are being brought in to do the other stuff, the stuff that allows everyone else to do what they need to do to have that organ functioning. And so they come in waves. So in fact, what we have right now in our lab are MIT88 knockout IL-10 reporter mice, because we want to show that it's not that the monocyte gets hit by things to express it, but it's the environment that's dictating their reparative function or their anti-inflammatory property. And not in a MIT88 knockout, but again in these chimeric models, right? Because if you did it straight up in a MIT88 knockout, then the environment itself won't sense the TLR ligand that you're giving, right? So that's why people don't see it. They just assume that monocytes can't respond, but it's because they're doing it in a straight up MIT88 knockout. And that, then that doesn't allow the environment to react to that either pathogen or TLR ligand. Yeah.
Bone marrow chimera was a great slide that inspires students in faculty.
Thanks.
My question is a specific concept of, so my, hello, check. Thank you, Galactic Overlord. My question is about on a metastasis creating localized immunity, and then in order to get to a metastatic niche, it can't because it hasn't established systemic immunity. It has localized immunity in that tissue. So I'm thinking there's an experiment or a model here where these monocytes are going to be coming and the tumor has to recruit them so that they can promote systemic immunity at a pre-metastatic niche site, and that this bone marrow chimera experiment, instead of doing sort of an early fate mapping, could be timed out so that you could see this progression when you injected your tumor, establishing localized immunity at a primary or metastatic site to progress to a secondary site. Does that make sense of sort of my brain just sort of, this is on the spot stream of consciousness from this experiment, but I mean, this is how I'm seeing this model of your barriers to metastatic colonization to distant tissue sites. You have a tissue monocyte, right, at a foreign site. So let's say you're in the breast or the prostate and you want to get to the bone. How do you get to those access to those monocytes so that you can suppress T cells, so you can maintain a new niche, right, to get to that site? Because right now, those monocytes are not suppressing the T cell recognition of that tumor, right? So I'm sure there's an IL-10 dependent mechanism in there that the tumors are indirectly getting those monocytes to produce at that site. Is that, sorry, maybe a little too much?
Maybe you guys can chat about that afterwards.
Sorry.
Yeah, but I wanted to just ask one more thing since the cancer biologist is here. So how much is known about what's recruited, these myeloid suppressor cells? I mean, people know that they derive from monocytes, but how much is there versus what's recruited in? I mean, can people distinguish the resident versus the infiltrating macrophages in the tumor microenvironment?
I know from my models in pancreas, breast, prostate, that we can see this orderly progressive flow of the localized MDSC versus the splenic derived. It's sort of a pooling cascade, and then we can see if we can let our models, our criterias go longer depending on them, we can have a bone marrow derived MDSC. Okay. And that's phenotypically Dmitry Gabrilovich, all those guys, I mean, that, I think there's at least 117 markers, right, that you can okay. make an argument for that population. But it is definitively different between that tissue MDSC versus the splenic derived.
Okay.